History of TCA cycle. The history of the tricarboxylic acid TCA cycle, also known as the Krebs cycle or citric acid cycle, is a fascinating tale of scientific discovery and collaboration. Several researchers contributed to the understanding of the central metabolic pathway. Early pioneers, before 1937, scientists like Karl Marcias, Frey Knoop, and Albert Szent-Györgyi had elucidated many reactions now recognized as part of the TCA cycle. Their work laid the groundwork for the cycle's complete elucidation. Sir Huss Adolf Krebs. The complete cycle was proposed by Sir Huss Krebs in 1937. His work earned him the Nobel Prize in Physiology or Medicine in 1953. Krebs initially named the TCA tricarboxylic acid cycle because he was uncertain if citric acid was a member. He submitted his findings to the journal Nature, but it was rejected. He eventually published it in the journal Enzymologia. Krebs' discovery of the cycle built upon his earlier experience characterizing the urea cycle. He identified that malonate, a succinate dehydrogenase inhibitor, blocked pyruvate metabolism, leading to the accumulation of succinate, citrate, and alpha-ketoglutarate. This suggested that succinate was a product of pyruvate metabolism and that tricarboxylic acids were intermediates. His observations and understanding of metabolic cycles led him to propose the TCA cycle. W. A. Johnson. In 1937, Krebs and W. A. Johnson collaborated on a groundbreaking paper titled The Role of Citric Acid in Intermediate Metabolism in Animal Tissues, solidifying the importance of citric acid in the cycle. PDF. A. G. Ogston. Later, in 1948, A. G. Ogston demonstrated that the tricarboxylic acid in question was indeed citric acid, leading to the name citric acid cycle. Carl Wilhelm Scheele had isolated citric acid from citrus fruits back in 1780. Beyond Krebs, subsequent research by numerous scientists further elaborated on the TCA cycle's intricacies, regulation, and connections to other metabolic pathways. This included studies on the pyruvate dehydrogenase complex, the entry point for carbohydrates into the cycle, and anaplerotic reactions, which replenish cycle intermediates. The history of the TCA cycle demonstrates the collaborative nature of scientific discovery. From the early pioneers to Krebs' pivotal contribution and the subsequent refinements by countless researchers, the understanding of this crucial metabolic pathway has evolved through decades of dedicated research. The TCA cycle has multiple names, each reflecting different aspects of this crucial metabolic pathway. Tricarboxylic acid TCA cycle. This name originates from the fact that the first product formed in the cycle, citrate, and the subsequent intermediate, isocitrate, are both tricarboxylic acids. This nomenclature highlights the chemical nature of key intermediates in the cycle. Krebs cycle. The cycle is named after Sir Huss Adolf Krebs, a British biochemist who made significant contributions to its elucidation in the 1930s. This name honors the scientist who first postulated the cycle. Citric acid cycle. Citric acid is the first intermediate formed in the cycle through the condensation of oxaloacetate and acetyl-CoA. This name emphasizes the starting point and the cyclic nature of the pathway. All three names are widely used and accepted in the scientific community, and they all refer to the same fundamental metabolic pathway responsible for the oxidation of acetyl-CoA and energy production in the form of ATP.
Summary of TCA cycle. The TCA cycle, also known as the Krebs cycle or citric acid cycle, is a vital metabolic pathway located in the mitochondrial matrix of eukaryotic cells. This cycle serves as the central hub for the oxidation of carbohydrates, fats, and proteins, with their metabolic breakdown products converging to form acetyl-CoA, the primary substrate for the TCA cycle. The cycle's primary function is to generate energy in the form of ADP through oxidative phosphorylation. The TCA cycle consists of 8 sequential enzymatic reactions. Citrate synthase catalyzes the condensation of acetyl-CoA 2C with oxaloacetate for C to form citrate 6C. This is an irreversible step. Aconitase converts citrate to isocitrate via cisaconitase. Isocitrate dehydrogenase catalyzes the oxidative decarboxylation of isocitrate to alpha-ketoglutarate, 5C, producing NADH and CO2. Alpha-ketoglutarate dehydrogenase complex carries out the oxidative decarboxylation of alpha-ketoglutarate to succinyl-CoA for C, generating NADH and CO2. Succinyl-CoA synthetase, succinate thiokinase, converts succinyl-CoA to succinate, producing GTP through substrate-level phosphorylation. Succinate dehydrogenase oxidizes succinate to fumarate, producing FADH2. This enzyme is located in the inner mitochondrial membrane. Fumarase hydrates fumarate to form L-malate. Malate dehydrogenase oxidizes malate to oxaloacetate, generating NADH. Energy yield. Each turn of the TCA cycle, fueled by one molecule of acetyl-CoA, yields 3 NADH molecules, equivalent to 7.5 ADP through oxidative phosphorylation. 1 FADH2 molecule, equivalent to 1.5 ADP. 1 GTP molecule, equivalent to 1 ADP. This results in a total of 10 ADP molecules generated per acetyl-CoA molecule oxidized. Regulation. The TCA cycle is tightly regulated to match the energy requirements of the cell. Key regulatory points include pyruvate dehydrogenase complex, PDHC, allosterically inhibited by NADH, acetyl-CoA, and ADP. Allosterically activated by pyruvate, ADP, and CoA-SH. Inactivated by phosphorylation via PDH kinase, which is activated by high-energy signals. Activated by dephosphorylation via PDH phosphatase, which is activated by insulin and Ca2+. Citrate synthase, inhibited by ADP, citrate, and long-chain acyl-CoA. Isocitrate dehydrogenase, inhibited by ADP and NADH, activated by ADP and Ca2+. Alpha-ketoglutarate dehydrogenase complex, inhibited by NADH and succinyl-CoA, activated by Ca2+. Additionally, substrate availability, particularly oxaloacetate and acetyl-CoA, and the cellular redox state, NAD+/NADH ratio, play crucial roles in regulating the TCA cycle. Amphibolic nature. The TCA cycle is amphibolic, meaning it participates in both catabolic and anabolic processes. While it serves as the final oxidative pathway for energy production, its intermediates are also precursors for various biosynthetic pathways, including gluconeogenesis, synthesis of glucose from non-carbohydrate sources, fatty acid synthesis, amino acid synthesis, heme synthesis for hemoglobin and other hemoproteins. Anaplerotic reactions replenish TCA cycle intermediates that are removed for biosynthesis. The pyruvate carboxylase reaction, converting pyruvate to oxaloacetate, is a prominent example of an anaplerotic reaction. In conclusion, the TCA cycle is a fundamental metabolic pathway crucial for energy production, biosynthesis, and cellular homeostasis. Its regulation is multifaceted and intricately linked to the energy status and metabolic needs of the cell, highlighting the complex interplay between catabolic and anabolic processes in living organisms. Step 1. Condensation of acetyl-CoA 2C with oxaloacetate 4C to form citrate 6C. Acetyl-CoA plus oxaloacetate plus H2O citrate plus CoA. Citrate synthase. The first step in the TCA cycle involves the condensation of acetyl-CoA, a 2-carbon molecule, with oxaloacetate, a 4-carbon molecule, to form citrate, a 6-carbon molecule. This reaction is catalyzed by the enzyme citrate synthase. The reaction is driven by the cleavage of the high-energy thioester bond of citryl-CoA, an intermediate in the reaction. This reaction is irreversible and exothermic. The entry of acetyl-CoA into the TCA cycle via this reaction does not lead to the net production or consumption of intermediates. This is because the oxaloacetate used in the first step is regenerated upon completion of the cycle 4. The overall reaction is acetyl-CoA plus oxaloacetate plus H2O citrate plus CoA. The rate of this reaction, which is the most important regulatory step in the TCA cycle, is determined by several factors. These factors include the availability of substrates, allosteric modulation, and feedback inhibition. For instance, high levels of ADP and NADH inhibit citrate synthase, indicating a sufficient energy supply for the cell. High levels of ADP and AMP activate citrate synthase, signaling a need for more energy production. Succinyl-CoA, an intermediate of the TCA cycle, competes with acetyl-CoA and inhibits citrate synthase. Citrate, the product of the reaction, also acts as an inhibitor of citrate synthase. This regulation ensures that the TCA cycle operates efficiently based on the cell's energy requirements. Step 2. Isomerization of citrate to isocitrate. Citrate cisaconitate isocitrate. Aconitase. The second step of the TCA cycle involves the isomerization of citrate to isocitrate. This is a two-step reaction catalyzed by the enzyme aconitase. First, citrate is dehydrated to form cisaconitate. Then, cisaconitate is hydrated to form isocitrate. Aconitase is an iron-sulfur protein that requires Fe2+ for its activity. The reaction is reversible, and the equilibrium favors the formation of citrate. However, the reaction proceeds towards the formation of isocitrate due to the rapid removal of isocitrate in the subsequent step of the cycle. The overall reaction is citrate cisaconitate isocitrate. The conversion of citrate to isocitrate is necessary because citrate is a poor substrate for oxidation. In contrast, isocitrate has a secondary alcohol group that can be readily oxidized. This isomerization step sets the stage for the subsequent oxidative decarboxylation reaction in the TCA cycle. Step 3. Conversion of isocitrate to alpha-ketoglutarate. Isocitrate plus NAD plus alpha-ketoglutarate plus CO2 plus NADH plus H plus. Isocitrate dehydrogenase. The third step of the TCA cycle is the conversion of isocitrate to alpha-ketoglutarate. This is an oxidative decarboxylation reaction that is catalyzed by isocitrate dehydrogenase. The sources provide the following details for this step. Isocitrate dehydrogenase catalyzes the oxidation of isocitrate to oxalosuccinate, an unstable intermediate. Oxalosuccinate is decarboxylated by isocitrate dehydrogenase to produce alpha-ketoglutarate. This reaction releases the first CO2 molecule generated in the TCA cycle. This step generates the first NADH molecule from acetyl-CoA in the cycle. Overall reaction: isocitrate plus NAD plus alpha-ketoglutarate plus CO2 plus NADH plus H plus. The two carbon atoms that enter the cycle as acetyl-CoA are not lost as CO2 in this step. These carbon atoms will become part of the symmetric 4-carbon succinate molecule and will be lost as CO2 in subsequent cycles. Isocitrate dehydrogenase as a key regulatory enzyme of the TCA cycle. It is allosterically activated by ADP and Ca2+ and inhibited by ADP and NADH. The inhibition of isocitrate dehydrogenase by ADP and NADH allows the TCA cycle to be regulated by the energy status of the cell. When energy levels are high, the cycle is slowed down. When energy levels are low, the cycle is sped up. Step 4. Conversion of alpha-ketoglutarate to succinyl-CoA. Alpha-ketoglutarate plus CoA plus NAD plus succinyl-CoA plus CO2 plus NADH plus H plus. Ketoglutarate dehydrogenase complex. The fourth step of the TCA cycle is the conversion of alpha-ketoglutarate to succinyl-CoA. This is also an oxidative decarboxylation reaction, and it is catalyzed by the alpha-ketoglutarate dehydrogenase complex. This complex is similar in mechanism, cofactors, and coenzymes to the pyruvate dehydrogenase complex. The reaction produces the second CO2 molecule generated in the TCA cycle and the second NADH molecule from acetyl-CoA in the cycle. The sources provide the following details. Alpha-ketoglutarate and CoA come together, and in the process, a molecule of carbon dioxide is released, and NAD+ is reduced to NADH. This is the second and last carbon loss from acetyl-CoA in the cycle. Overall reaction: alpha-ketoglutarate plus CoA plus NAD plus succinyl-CoA plus CO2 plus NADH plus H plus. The alpha-ketoglutarate dehydrogenase complex is inhibited by NADH and succinyl-CoA and activated by Ca2 plus. This reaction is not readily reversible because of the decarboxylation step. This step is important for regulating the overall rate of the TCA cycle. The two carbons from the acetyl-CoA molecule that entered the cycle earlier are now completely removed. Step 5. Conversion of succinyl-CoA to succinate. Succinyl-CoA plus GDP plus pi, succinate plus GTP plus CoASH. Succinyl-CoA synthetase, succinate thiokinase. The fifth step of the TCA cycle is the conversion of succinyl-CoA to succinate. This reaction is a substrate-level phosphorylation, in which the energy released from the hydrolysis of the thioester bond in succinyl-CoA is used to generate a molecule of GTP or ADP. It is catalyzed by succinyl-CoA synthetase, also known as succinate thiokinase. The sources provide the following details for this step. Succinyl-CoA is a high-energy compound, similar to acetyl-CoA. The thioester bond of succinyl-CoA is cleaved, and the energy released is used to drive the synthesis of GTP or ADP. Overall reaction: succinyl-CoA plus GDP plus pi succinate plus GTP plus CoASH. In some tissues like liver and kidney, the enzyme produces GTP, but the GTP is energetically equivalent to ADP and can be readily converted to ADP by the enzyme nucleoside diphosphate kinase. This is the only example of substrate-level phosphorylation in the TCA cycle. This step is important for linking the TCA cycle to the electron transport chain, as the GTP or ADP produced in this step can be used to power the synthesis of ADP by oxidative phosphorylation. This reaction is reversible. Step 6. Conversion of succinate to fumarate. Succinate plus FAD fumarate plus FADH2. Succinate dehydrogenase. The sixth step of the TCA cycle is the conversion of succinate to fumarate. This is an oxidation reaction in which two hydrogens are removed from succinate by succinate dehydrogenase, reducing FAD to FADH2. After this step, 6 of the 8 electrons from the carbons in acetyl-CoA have been captured. Overall reaction: succinate plus FAD fumarate plus FADH2. Here are some key details for this step. Succinate dehydrogenase is the only enzyme of the TCA cycle embedded in the inner mitochondrial membrane. All other enzymes of the cycle are in the matrix. FADH2 produced in this reaction transfers electrons directly to the electron transport chain, as succinate dehydrogenase is a part of complex 2 of the electron transport chain. This reaction is reversible. The sources do not specify any regulatory mechanism for succinate dehydrogenase. This step is important because it links the TCA cycle to the electron transport chain, and it produces FADH2, which is used to generate ADP by oxidative phosphorylation. Step 7. Conversion of fumarate to malate. Fumarate plus H2O malate. Fumarase, fumarate hydratase. The seventh step of the TCA cycle is the conversion of fumarate to malate. This is a hydration reaction in which a water molecule is added across the double bond of fumarate by the enzyme fumarase, also called fumarate hydratase. Overall reaction: fumarate plus H2O malate. The sources state the following. The reaction is reversible, but always proceeds unidirectionally toward the formation of malate due to the thermodynamic pull in the TCA cycle. This step is important because it prepares the molecule for the final oxidation step of the cycle. Step 8. Conversion of malate to oxaloacetate. Malate plus NAD plus oxaloacetate plus NADH plus H plus. Malate dehydrogenase. The eighth and final step of the TCA cycle is the conversion of malate to oxaloacetate. This reaction is catalyzed by malate dehydrogenase and is coupled to the reduction of NAD plus to NADH, capturing the last 2 electrons from the original acetyl-CoA molecule. The oxaloacetate produced in this step can then condense with another molecule of acetyl-CoA to initiate another cycle. Overall reaction: malate plus NAD plus oxaloacetate plus NADH plus H+. Here are some important details for this step. This is the fourth and final oxidation reaction in the TCA cycle, generating the third NADH molecule of the cycle. Even though the reaction is reversible, it proceeds in the forward direction because of the highly exergonic citrate synthase reaction, the first step of the cycle, which quickly uses up the oxaloacetate. The sources do not mention any regulatory mechanisms for malate dehydrogenase. This step is important because it regenerates oxaloacetate, the starting molecule of the TCA cycle, allowing the cycle to continue. This step marks the completion of one full turn of the TCA cycle, during which one molecule of acetyl-CoA has been completely oxidized to two molecules of CO2. In the process, the cycle has produced three molecules of NADH, one molecule of FADH2, and one molecule of GTP, or ADP, which can be used to generate ADP through oxidative phosphorylation. Energetics: the tricarboxylic acid TCA cycle, also called the Krebs cycle or citric acid cycle, is a crucial metabolic pathway that takes place in the mitochondria of eukaryotic cells. It is the final common pathway for the oxidation of carbohydrates, fats, and proteins, and it plays a central role in energy production and biosynthesis. The cycle involves a series of 8 enzymatic reactions that oxidize acetyl-CoA, derived from various metabolic fuels, to carbon dioxide, CO2, producing reduced coenzymes, NADH and FADH2, and ADP. Here is a detailed account of the energetics of the TCA cycle, focusing on the production, utilization, and net energy yield from the substrate acetyl-CoA. Energy production: the TCA cycle generates energy through oxidative phosphorylation, which involves the transfer of electrons from reduced coenzymes, NADH and FADH2, to molecular oxygen through the electron transport chain, ultimately producing ADP. Substrate-level phosphorylation also contributes to ADP production in the cycle. Substrate: the primary substrate for the TCA cycle is acetyl-CoA, a 2-carbon molecule derived from the breakdown of carbohydrates, fatty acids, and amino acids. Each molecule of acetyl-CoA enters the cycle and is completely oxidized to 2 molecules of CO2. Energy yield from one acetyl-CoA molecule. The following table summarizes the ADP production steps in the TCA cycle. Step reaction: coenzyme, reduced ADP yield. 3. Isocitrate to alpha-ketoglutarate isocitrate plus NAD plus alpha-ketoglutarate plus CO2 plus NADH plus H plus NADH 2.5. 4. Alpha-ketoglutarate to succinyl-CoA alpha-ketoglutarate plus CoA plus NAD plus succinyl-CoA plus CO2 plus NADH plus H plus NADH 2.5. 5. Succinyl-CoA to succinate succinyl-CoA plus GDP plus pi succinate plus GTP plus CoSH1. 6. Succinate to fumarate succinate plus FAD fumarate plus FADH2 FADH2 1.5. 8. Malate to oxaloacetate malate plus NAD plus oxaloacetate plus NADH plus H plus NADH 2.5. Total per acetyl-CoA molecule: 10. Net energy yield from one glucose molecule. One glucose molecule undergoes glycolysis to produce 2 pyruvate molecules. Each pyruvate molecule is converted to one acetyl-CoA molecule through oxidative decarboxylation, yielding one NADH molecule. Therefore, one glucose molecule generates 2 acetyl-CoA molecules, which enter the TCA cycle. The overall energy yield from the complete oxidation of one glucose molecule in the TCA cycle, including the contribution from glycolysis and pyruvate decarboxylation, is as follows. Glycolysis: 2 ADP, net, plus 2 NADH, 5 ADP, equals 7 ADP. Pyruvate decarboxylation: 2 NADH, 5 ADP, equals 5 ADP. TCA cycle: 2 acetyl-CoA times 10 ADP slash acetyl-CoA equals 20 ADP. Total ADP yield per glucose molecule: 7 ADP plus 5 ADP plus 20 ADP equals 32 ADP. Utilization of energy. The ADP generated from the TCA cycle is used to power various cellular processes, such as muscle contraction, active transport, biosynthesis, and signal transduction. The reduced coenzymes, NADH and FADH2, produced in the cycle serve as electron carriers, transferring electrons to the electron transport chain, which ultimately drives ADP synthesis. Regulation. The TCA cycle is tightly regulated to match the energy demands of the cell. Key enzymes in the cycle, such as citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase, are regulated by substrate availability, allosteric effectors, and feedback inhibition. Key concepts. The TCA cycle is a central hub of metabolism, connecting carbohydrate, lipid, and protein metabolism. It is an amphibolic pathway, meaning it has both catabolic, energy-producing, and anabolic, biosynthetic functions. The cycle generates reduced coenzymes, which are essential for ADP production through oxidative phosphorylation. The energetics of the TCA cycle illustrate the intricate and efficient mechanisms of energy production in cells. The TCA cycle is a vital metabolic pathway that is essential for the survival of eukaryotic organisms. Understanding its energetics provides insights into how cells generate energy from various metabolic fuels and how this energy is used to sustain life. Regulation of TCA cycle. The tricarboxylic acid TCA cycle, also known as the Krebs cycle or citric acid cycle, is a crucial metabolic pathway that plays a central role in energy production and biosynthesis. The cycle's activity is tightly regulated to meet the cell's energy demands and to ensure efficient utilization of metabolic fuels. Regulation occurs at multiple levels, including 1. Regulation of pyruvate dehydrogenase complex. The pyruvate dehydrogenase complex, PDHC, links glycolysis to the TCA cycle by converting pyruvate to acetyl-CoA, the primary substrate for the cycle. The activity of PDHC is regulated by both allosteric and covalent modifications. Allosteric regulation. Inhibitors. NADH, acetyl-CoA, and ADP inhibit PDHC activity. These molecules signal a high-energy state in the cell, indicating that there is sufficient energy and the cycle can slow down. Activators. Pyruvate, ADP, and CoA-SH activate PDHC. These molecules indicate a low-energy state, signaling the need for increased energy production through the TCA cycle. Covalent modification, phosphorylation/dephosphorylation. PDH kinase phosphorylates and inactivates PDHC. The kinase is activated by NADH, acetyl-CoA, and ADP, high-energy signals. PDH phosphatase dephosphorylates and activates PDHC. The phosphatase is activated by insulin and CA2+, signals for increased glucose utilization and muscle contraction, respectively. 2. Regulation of TCA cycle enzymes. Several key enzymes within the TCA cycle are also regulated, ensuring that the cycle's activity is finely tuned to the cell's energy needs. Citrate synthase catalyzes the first step of the cycle, condensation of oxaloacetate and acetyl-CoA to form citrate. Inhibited by ADP, citrate, its product, and long-chain acyl-CoA. This feedback inhibition prevents the accumulation of citrate when energy levels are high or when alternative fuels, fatty acids, are available. Isocitrate dehydrogenase catalyzes the third step of the cycle, oxidative decarboxylation of isocitrate to alpha-ketoglutarate. Inhibited by ADP and NADH, high-energy signals. Activated by ADP and CA2+, low-energy signals. Calcium activation couples energy production to muscle contraction. Alpha-ketoglutarate dehydrogenase complex catalyzes the fourth step of the cycle, oxidative decarboxylation of alpha-ketoglutarate to succinyl-CoA. Inhibited by NADH and succinyl-CoA, its product. This feedback inhibition prevents the buildup of intermediates when energy levels are high. Activated by CA2+. 3. Substrate availability. The rate of the TCA cycle is also influenced by the availability of substrates, particularly oxaloacetate and acetyl-CoA. Oxaloacetate. The concentration of oxaloacetate can limit the rate of citrate synthesis. Anaplerotic reactions, such as the pyruvate carboxylase reaction, replenish oxaloacetate levels. Acetyl-CoA. Acetyl-CoA is primarily derived from pyruvate, from glycolysis, or from fatty acid oxidation. Its availability can directly impact the rate of the cycle. 4. Redox state, NAD plus/NADH ratio. The activity of the TCA cycle depends on the availability of NAD plus for the dehydrogenase reactions. This is linked to the rate of NADH consumption by the electron transport chain, which is ultimately determined by the rate of ADP utilization and production of ADP. When ADP is utilized for metabolic work, ADP is produced, NADH is consumed by the electron transport system for ADP production, and NAD plus is regenerated. This activates the TCA cycle, leading to increased fuel consumption and NADH production to support further ADP synthesis. Conversely, high levels of NADH inhibit the TCA cycle. 5. Hormonal regulation. Hormonal signals can indirectly regulate the TCA cycle by influencing the activities of enzymes involved in fuel metabolism. Insulin stimulates pyruvate dehydrogenase by activating the phosphatase. This directs carbohydrate-derived carbons into fatty acid synthesis via citrate synthase. Glucagon promotes gluconeogenesis, leading to the diversion of TCA cycle intermediates towards glucose synthesis. Epinephrine, adrenaline promotes glycogenolysis, increasing the availability of glucose for glycolysis and subsequently acetyl-CoA for the TCA cycle. 6. Gene expression and enzyme synthesis. The levels of TCA cycle enzymes can be regulated at the level of gene expression and protein synthesis. Diet, for example, can control the expression of pyruvate dehydrogenase kinases, influencing the activity of the PDHC. Regulation at the genetic and transport levels is not fully understood. In summary, the regulation of the TCA cycle is a complex and dynamic process that involves multiple levels of control, ensuring that the cycle's activity is tightly coupled to the cell's energy demands and metabolic state. This intricate regulation is crucial for maintaining cellular homeostasis and supporting life processes. Anaplerotic reactions of TCA cycle. Anaplerotic reactions are crucial for replenishing the intermediates of the tricarboxylic acid, TCA, cycle, ensuring its continuous operation. These reactions are essential because TCA cycle intermediates are often siphoned off for various biosynthetic processes, potentially depleting the cycle and halting energy production. Here are some important anaplerotic reactions. Pyruvate carboxylase reaction. This is one of the most important anaplerotic reactions. It converts pyruvate to oxaloacetate, a key TCA cycle intermediate required for the condensation reaction with acetyl-CoA. Pyruvate carboxylase is activated by high concentrations of acetyl-CoA, indicating a need for more oxaloacetate to sustain the cycle. This reaction is crucial in maintaining an adequate supply of oxaloacetate for the citrate synthase reaction, especially when acetyl-CoA levels increase. The reaction requires biotin, ADP, Mg++, and acetyl-CoA as a positive modifier. Malic enzyme reaction. This reaction converts pyruvate to malate in the cytoplasm. Malate can then enter the mitochondria and be converted to oxaloacetate, contributing to the TCA cycle. Aspartate transamination. Aspartate can be transaminated to form oxaloacetate. Glutamate and glutamine metabolism. Glutamate and glutamine can yield alpha-ketoglutarate through the actions of glutaminase and glutamate dehydrogenase. Propionyl-CoA to succinyl-CoA. Propionyl-CoA, derived from the metabolism of odd-chain fatty acids and some amino acids, can be converted to succinyl-CoA, a TCA cycle intermediate. Importance of anaplerotic reactions. Maintaining TCA cycle function. Anaplerotic reactions ensure a constant supply of TCA cycle intermediates, preventing depletion due to their utilization in biosynthetic pathways. Balancing cataplerosis. Cataplerotic reactions remove intermediates from the TCA cycle for biosynthesis. Anaplerotic reactions counterbalance these removals, maintaining a steady state of intermediates within the cycle. Metabolic flexibility. They provide metabolic flexibility, allowing the cell to adapt to changes in nutrient availability and energy demands. Connecting metabolic pathways. Anaplerotic reactions link the TCA cycle to various other metabolic pathways, including glycolysis, gluconeogenesis, fatty acid metabolism, and amino acid metabolism. In essence, anaplerotic reactions are essential for the proper functioning of the TCA cycle, energy production, and the overall metabolic balance of the cell. They ensure that the TCA cycle can continue to operate efficiently, even when intermediates are diverted for other cellular processes. Cataplerotic reactions of TCA cycle. Cataplerotic reactions are metabolic pathways that utilize intermediates of the TCA cycle for biosynthetic purposes, effectively removing them from the cycle. These reactions are countered by anaplerotic reactions, which replenish the cycle intermediates to maintain a steady state. Here is a list of some important cataplerotic reactions. Citrate to fatty acid synthesis. Citrate, when in excess, can be transported out of the mitochondria to the cytoplasm. In the cytoplasm, it is cleaved by ADP citrate lyase to generate acetyl-CoA, the precursor for fatty acid synthesis. This process is particularly active in the liver in the well-fed state. Oxaloacetate to gluconeogenesis. Oxaloacetate can be converted to phosphoenol pyruvate, PEP, by PEP carboxykinase. PEP is a key intermediate in gluconeogenesis, the pathway that synthesizes glucose from non-carbohydrate precursors. This is crucial during fasting or starvation when glucose levels are low. Alpha-ketoglutarate to glutamate. Alpha-ketoglutarate can be transaminated to glutamate, a major excitatory neurotransmitter and a precursor for other amino acids like glutamine and proline. Oxaloacetate to aspartate. Similarly, oxaloacetate can be transaminated to aspartate, another non-essential amino acid that serves as a precursor for other biomolecules, including nucleotides. Succinyl-CoA to heme synthesis. Succinyl-CoA is a precursor for porphyrin synthesis, which ultimately leads to the formation of heme, the iron-containing prosthetic group in hemoglobin and other hemoproteins. The balance between cataplerotic and anaplerotic reactions is crucial for maintaining a functional TCA cycle. When cataplerotic reactions exceed anaplerotic reactions, the cycle can be depleted of intermediates, leading to reduced energy production and impaired cellular metabolism. Conversely, an excess of anaplerotic reactions can lead to an accumulation of TCA cycle intermediates, potentially disrupting metabolic balance. The regulation of these reactions ensures that the TCA cycle functions efficiently to meet the energy and biosynthetic demands of the cell. Amphibolic nature of TCA cycle. The TCA cycle, also known as the Krebs cycle or the citric acid cycle, is a central metabolic pathway in the mitochondrial matrix that plays a crucial role in both catabolism and anabolism. This dual functionality earns it the designation of an amphibolic pathway. Catabolic role. The primary catabolic function of the TCA cycle is to oxidize acetyl-CoA derived from carbohydrates, lipids, and proteins, ultimately yielding CO2, H2O, and energy in the form of ADP. This oxidation process generates reduced coenzymes, NADH and FADH2, which feed into the electron transport chain, driving oxidative phosphorylation for ADP production. Anabolic role. Beyond its catabolic role, the TCA cycle serves as a source of precursors for various biosynthetic pathways. Intermediates of the TCA cycle are siphoned off to participate in the synthesis of essential biomolecules. Glucose. During starvation and fasting, TCA cycle intermediates, such as oxaloacetate and malate, contribute to gluconeogenesis, the pathway that synthesizes glucose from non-carbohydrate sources. Fatty acids. Citrate, exported from the mitochondria, provides the cytoplasmic acetyl-CoA necessary for fatty acid synthesis. Amino acids. Oxaloacetate and alpha-ketoglutarate are precursors for the synthesis of the amino acids aspartate and glutamate, respectively. These amino acids, in turn, can be used to generate other non-essential amino acids. Heme, succinyl-CoA, generated in the TCA cycle, is a vital precursor for the synthesis of porphyrins, the building blocks of heme, essential for oxygen transport in hemoglobin. Regulation and balance. The amphibolic nature of the TCA cycle necessitates a delicate balance between its catabolic and anabolic functions. Anaplerotic reactions play a crucial role in replenishing TCA cycle intermediates that are diverted for biosynthetic purposes. The regulation of the TCA cycle is complex and involves substrate availability, allosteric effectors, and feedback inhibition to ensure efficient energy production and biosynthesis coordination. The TCA cycle is a prime example of the interconnectedness of metabolic pathways, serving as a central hub for energy generation and the biosynthesis of diverse biomolecules vital for cellular function. Coenzymes involved in TCA cycle. The tricarboxylic acid TCA cycle, also called the Krebs cycle or citric acid cycle, is a key metabolic pathway that oxidizes acetyl-CoA, which is derived from the breakdown of carbohydrates, lipids, and some amino acids. This oxidation generates energy, which is captured by reduced coenzymes NADH and FADH2, subsequently fueling the electron transport chain to produce ADP. Here is a detailed look at the coenzymes in the TCA cycle, the steps they are involved in, and the vitamins they are derived from. Thiamine pyrophosphate, TPP, derived from vitamin B1, thiamine. Steps involved. Conversion of pyruvate to acetyl-CoA. TPP is a crucial coenzyme for the pyruvate dehydrogenase complex, PDHC, which catalyzes the oxidative decarboxylation of pyruvate to acetyl-CoA. This is the link between glycolysis and the TCA cycle. Conversion of alpha-ketoglutarate to succinyl-CoA. TPP is also required by the alpha-ketoglutarate dehydrogenase complex, which catalyzes the oxidative decarboxylation of alpha-ketoglutarate to succinyl-CoA. This reaction releases CO2 and generates NADH. Lipoic acid, derived from, not derived from a vitamin. It is synthesized by the body. Step involved. Pyruvate dehydrogenase complex, PDHC lipoic acid functions as a carrier of both hydrogens and acetyl groups within the PDHC. It facilitates the transfer of the acetyl group from pyruvate to coenzyme A, forming acetyl-CoA. Flavin adenine dinucleotide, FAD, derived from vitamin B2, riboflavin. Step involved. Conversion of succinate to fumarate. FAD is an electron carrier involved in the oxidation of succinate to fumarate. This reaction is catalyzed by succinate dehydrogenase, which is embedded in the inner mitochondrial membrane and acts as complex 2 of the electron transport chain. Nicotinamide adenine dinucleotide, NAD+, derived from vitamin B3 niacin. Steps involved. Conversion of isocitrate to alpha-ketoglutarate. NAD+ acts as an electron acceptor in the conversion of isocitrate to alpha-ketoglutarate, catalyzed by isocitrate dehydrogenase. This reaction generates NADH and releases CO2. Conversion of alpha-ketoglutarate to succinyl-CoA. NAD+ is also involved in the oxidative decarboxylation of alpha-ketoglutarate to succinyl-CoA by the alpha-ketoglutarate dehydrogenase complex. This step generates NADH and releases CO2. Conversion of malate to oxaloacetate. NAD+ is required for the final step of the TCA cycle, where malate is oxidized to oxaloacetate by malate dehydrogenase. This reaction produces NADH, regenerating oxaloacetate for the next cycle. Coenzyme A, CoA, or CoSH, derived from vitamin B5, pantothenic acid. Steps involved. Entry of acetyl-CoA into the TCA cycle. CoA is an acyl group carrier that plays a crucial role in carrying the acetyl group into the cycle, initiating the condensation reaction with oxaloacetate to form citrate. Formation of succinyl-CoA. CoA is involved in the formation of succinyl-CoA from alpha-ketoglutarate. These coenzymes and their associated vitamins are essential for the TCA cycle's function, enabling complete acetyl-CoA oxidation and ADP production via oxidative phosphorylation. Disorders of the TCA cycle. Disorders of the TCA cycle are rare, but can have serious consequences due to the cycle's central role in energy production and biosynthesis. These disorders can be caused by genetic defects affecting the enzymes of the cycle or by factors that disrupt the cycle's function. Genetic defects: pyruvate dehydrogenase complex deficiency. A deficiency in the pyruvate dehydrogenase complex, PDHC, the entry point for carbohydrates into the TCA cycle, is the most common biochemical cause of congenital lactic acidosis. This deficiency leads to a buildup of pyruvate, which is converted to lactate, causing lactic acidosis. The brain, which relies heavily on the TCA cycle for energy, is particularly affected, leading to neurological problems. Deficiencies in TCA cycle enzymes. Germline mutations in TCA cycle enzymes are linked to distinct cancer subtypes. Succinate dehydrogenase subunit mutations. These can cause pheochromocytomas and paragangliomas. Fumarate hydratase mutations. These are associated with hereditary leiomyoma and renal cell carcinoma, HLRCC. Isocitrate dehydrogenase 1, IDH1 mutations. These are found in various glioma subtypes and lead to the production of 2-hydroxyglutarate from alpha-ketoglutarate, potentially contributing to tumor development. Factors disrupting TCA cycle function. Fluoroacetate poisoning. Fluoroacetate is converted to fluoroacetate, a potent inhibitor of aconitase. This inhibition blocks the TCA cycle, leading to reduced energy production and cell death. Arsenic poisoning. Arsenic inhibits the alpha-ketoglutarate dehydrogenase complex, leading to the accumulation of alpha-ketoglutarate and disrupting the cycle. Malonate inhibition. Malonate is a competitive inhibitor of succinate dehydrogenase, blocking the conversion of succinate to fumarate and disrupting the cycle. Metabolic disorders. Conditions like diabetes mellitus can indirectly affect the TCA cycle. In diabetes, the inability to properly utilize glucose can lead to an accumulation of acetyl-CoA from fatty acid oxidation. This excess acetyl-CoA, in the absence of sufficient oxaloacetate, can be diverted towards ketone body formation, leading to ketoacidosis. Consequences of TCA cycle disorders. Energy depletion. Impaired TCA cycle function reduces ADP production, leading to energy deficiency in cells and tissues. Lactic acidosis. A buildup of pyruvate due to TCA cycle dysfunction leads to increased lactate production, causing lactic acidosis. Neurological dysfunction. The brain's high energy demand makes it particularly vulnerable to TCA cycle defects, resulting in neurological symptoms. Metabolic imbalances. Disruption of the TCA cycle affects various biosynthetic pathways, leading to imbalances in amino acid, glucose, and lipid metabolism. Diagnosis and treatment. Diagnosis of TCA cycle disorders involves a combination of clinical evaluation, biochemical testing, measuring levels of specific metabolites, and genetic testing. Treatment varies depending on the specific disorder and may include dietary modifications. For certain disorders, specific dietary restrictions or supplements may help manage symptoms. Cofactor supplementation. Some enzyme deficiencies can be partially compensated by providing the necessary cofactors like thiamine. Enzyme replacement therapy. In some cases, enzyme replacement therapy may be an option, but this is still under development. Overall, understanding the causes and consequences of TCA cycle disorders is crucial for developing effective diagnostic and therapeutic strategies. These disorders highlight the importance of the TCA cycle in maintaining cellular energy balance and metabolic homeostasis. Genetic mutations of the TCA cycle enzymes. Disruptions in the TCA cycle, mainly due to mutations in genes encoding TCA cycle enzymes, can lead to various disorders, including cancer and metabolic diseases. Key TCA cycle enzyme defects and associated disorders. Isocitrate dehydrogenase, IDH mutations. Mutations in IDH1 and IDH2 genes are implicated in the development of gliomas and other cancers. These mutations result in the production of 2-hydroxyglutarate, 2-HG, instead of alpha-ketoglutarate. 2-HG acts as an oncometabolite, inhibiting alpha-ketoglutarate-dependent dioxygenases involved in various cellular processes, including DNA and histone demethylation, leading to altered gene expression and contributing to cancer development. Succinate dehydrogenase (SDH) mutations. Mutations in genes encoding the four subunits of the SDH complex, SDHA, SDHB, SDHC, and SDHD, are linked to familial cancer syndromes, including paragangliomas, pheochromocytomas, gastrointestinal stromal tumors, and renal cell carcinoma. SDH mutations result in succinate accumulation, leading to a pseudohypoxic state characterized by the activation of the hypoxia-inducible factor 1, HIF1, pathway and increased angiogenesis, promoting tumor growth. Fumarate hydratase, FH, mutations. FH gene mutations are associated with cancers such as cutaneous and uterine leiomyomas and renal cell carcinoma. Fumarate accumulation in FH-deficient cells inhibits alpha-ketoglutarate-dependent dioxygenases, including those involved in HIF1 regulation, contributing to cancer development. Pyruvate dehydrogenase complex, PDHC deficiency. PDHC deficiency, largely due to mutations in the PDHA1 gene, impairs the conversion of pyruvate to acetyl-CoA, a crucial step for TCA cycle entry. This deficiency leads to the accumulation of pyruvate and lactate, resulting in lactic acidosis, particularly affecting highly aerobic tissues like the brain, skeletal muscle, and heart. PDHC deficiency manifests as severe cerebral lactic acidosis and neurological deficiencies shortly after birth. Role of the TCA cycle in inflammation. The TCA cycle also plays a role in inflammatory responses. Itaconate, produced from cisaconate by aconate decarboxylase 1, encoded by the ACOD1 gene, accumulates in activated macrophages. Itaconate exerts anti-inflammatory effects by inhibiting SDH, leading to reduced ROS production. Decreased ROS levels allow prolyl hydroxylase domain, PHD, enzymes to hydroxylate HIF1 alpha, targeting it for proteasomal degradation. This, in turn, suppresses the production of pro-inflammatory cytokines such as IL-1 beta. Synthesis of acetyl-CoA or sources. The synthesis of acetyl-CoA is a crucial step in cellular metabolism, serving as a central link between carbohydrate, lipid, and protein metabolism. Its primary function is to deliver the acetyl group to the citric acid cycle, TCA cycle, for energy production. Here's a detailed explanation of its synthesis. 1. From pyruvate, carbohydrate metabolism. The most common route for acetyl-CoA production is through the oxidative decarboxylation of pyruvate. Pyruvate, the end product of glycolysis, is transported into the mitochondria. Inside the mitochondria, the pyruvate dehydrogenase complex, PDHC, catalyzes a multi-step reaction, converting pyruvate to acetyl-CoA. This complex is a multi-enzyme complex containing 3 enzymes. Pyruvate decarboxylase removes a carboxyl group from pyruvate, releasing CO2. Dihydrolipoyl transacetylase transfers the acetyl group to coenzyme A, forming acetyl-CoA. Dihydrolipoyl dehydrogenase reoxidizes the lipoamide cofactor using FAD and NAD+. 2. From fatty acids, lipid metabolism. Fatty acids, stored as triglycerides, are broken down into acetyl-CoA molecules through a process called beta oxidation. This process occurs in the mitochondria and involves a series of reactions that sequentially remove 2 carbon units from the fatty acid chain, each unit forming an acetyl-CoA molecule. Each cycle of beta oxidation produces 1 molecule of acetyl-CoA, FADH2, and NADH, which can enter the electron transport chain to generate ADP. 3. From amino acids, protein metabolism. Certain amino acids, known as ketogenic amino acids, can be broken down into acetyl-CoA or acetoacetyl-CoA, which can be further converted to acetyl-CoA. Examples of ketogenic amino acids include leucine, lysine, and tryptophan. 4. From ketone bodies. Ketone bodies, produced in the liver during periods of prolonged fasting or starvation, can be utilized by extrahepatic tissues, including the brain, as an energy source. Acetoacetate, a ketone body, can be converted back to acetoacetyl-CoA and then to acetyl-CoA, entering the TCA cycle. Regulation of acetyl-CoA synthesis. The synthesis of acetyl-CoA is tightly regulated, primarily at the level of the pyruvate dehydrogenase complex. Factors influencing this regulation include substrate availability. The availability of pyruvate, fatty acids, and amino acids influences the rate of acetyl-CoA production. Hormonal regulation. Insulin promotes glucose utilization and acetyl-CoA formation, while glucagon favors gluconeogenesis and inhibits PDHC activity. Allosteric regulation. Acetyl-CoA itself can inhibit PDHC, while pyruvate and CoA are positive allosteric regulators. Key points to remember. Acetyl-CoA is a central molecule in metabolism, connecting carbohydrate, lipid, and protein catabolic pathways. Its primary role is to fuel the TCA cycle for energy production. It's also a precursor for various biosynthetic pathways, including fatty acid and cholesterol synthesis. The regulation of acetyl-CoA synthesis is crucial for maintaining cellular energy balance and metabolic homeostasis. Overall, the synthesis of acetyl-CoA is a vital process in cellular metabolism. Its diverse sources and tight regulation highlight its importance in coordinating various metabolic pathways and ensuring the cell's energy needs are met under different physiological conditions. The pyruvate dehydrogenase complex, PDHC. The pyruvate dehydrogenase complex, PDHC, is a multi-enzyme complex that plays a critical role in cellular respiration, linking glycolysis to the citric acid cycle. This complex catalyzes the irreversible oxidative decarboxylation of pyruvate, the end product of glycolysis, to form acetyl-CoA, which then enters the citric acid cycle for further oxidation. Structural organization. The PDHC is a large macromolecular assembly located in the mitochondrial matrix of eukaryotes and in the cytoplasm of prokaryotes. It consists of multiple copies of three enzymatic subunits: E1, pyruvate dehydrogenase, E2, dihydrolipoyl transacetylase, and E3, dihydrolipoyl dehydrogenase. The core of the complex is formed by E2, arranged in a cubic or dodecahedral shape. E1 and E3 are arranged around the E2 core. Enzymatic activities and coenzymes. The PDHC requires 5 coenzymes for its activity. Thiamine pyrophosphate, TPP bound to E1. It decarboxylates pyruvate. Lipoic acid, covalently attached to E2. It acts as a swinging arm to transfer the acetyl group from E1 to CoA. Coenzyme A, CoA, accepts the acetyl group from lipoamide, forming acetyl-CoA. Flavin adenine dinucleotide, FAD bound to E3. It reoxidizes lipoamide. Nicotinamide adenine dinucleotide, NAD+ accepts electrons from FADH2, regenerating FAD. Reaction mechanism. Decarboxylation of pyruvate. Pyruvate dehydrogenase, E1, decarboxylates pyruvate, using TPP as a cofactor, forming hydroxyethyl TPP. CO2 is released in this step. Transfer of acetyl group to lipoamide. The hydroxyethyl group is oxidized to an acetyl group and transferred to lipoamide, forming acetyl dihydrolipoamide. Transacetylation. Dihydrolipoyl transacetylase, E2, transfers the acetyl group from lipoamide to CoA, forming acetyl-CoA. Reoxidation of lipoamide. Dihydrolipoyl dehydrogenase, E3, reoxidizes dihydrolipoamide, using FAD as a cofactor, forming FADH2. Electron transfer to NAD+. FADH2 is reoxidized by NAD+, forming NADH and H+, and regenerating FAD. Regulation of PDHC activity. The PDHC is tightly regulated to control the flow of carbon from glycolysis into the TCA cycle. Allosteric regulation. Inhibited by its products, acetyl-CoA and NADH. Activated by CoA, NAD+, and AMP. Covalent modification. Phosphorylation/dephosphorylation. In mammals, the E1 subunit is regulated by phosphorylation, inactivation, by pyruvate dehydrogenase kinase, PDK, and dephosphorylation, activation. by pyruvate dehydrogenase phosphatase, PDP. PDK is activated by high ADP, acetyl-CoA, and NADH levels. PDP is activated by Ca2+, and insulin. Clinical significance. PDHC deficiency. Deficiency of the PDHC is a rare but serious genetic disorder that can cause congenital lactic acidosis. This condition leads to a buildup of pyruvate, which is converted to lactate, causing lactic acidosis. The brain is particularly affected due to its high energy demand, resulting in neurological problems. Arsenic poisoning. Arsenic can inhibit the PDHC, leading to impaired energy production and cell death. The pyruvate dehydrogenase complex is a vital enzyme complex that plays a crucial role in cellular energy metabolism. Its intricate structure, complex reaction mechanism, and tight regulation highlight its significance in coordinating metabolic pathways and ensuring proper cellular function. Defects in the PDHC or disruption of its activity can lead to severe metabolic disorders. Transamination and TCA cycle. The tricarboxylic acid, TCA cycle, also known as the Krebs cycle or citric acid cycle, is a crucial metabolic pathway located in the mitochondria. It serves as a central hub for the oxidation of acetyl-CoA, which is derived from carbohydrates, lipids, and amino acids. This oxidation process generates energy in the form of ADP and also produces precursors for various biosynthetic pathways. Transamination is a reversible reaction that plays a vital role in connecting amino acid metabolism with the TCA cycle. Transamination involves the transfer of an amino group from an amino acid to a keto acid, catalyzed by enzymes called transaminases or aminotransferases. The most common keto acid acceptor in these reactions is alpha-ketoglutarate, a key intermediate of the TCA cycle. When an amino acid undergoes transamination with alpha-ketoglutarate, it forms glutamate, while the original keto acid is converted to its corresponding amino acid. This process effectively links amino acid metabolism to the TCA cycle. Here's a detailed explanation of the connection between transamination and the TCA cycle. Amino acids as energy sources. Transamination allows amino acids to contribute to energy production by converting them to keto acids, which can then enter the TCA cycle and be oxidized to generate ADP. Synthesis of non-essential amino acids. The TCA cycle provides a source of carbon skeletons for the synthesis of non-essential amino acids. The keto acids generated in the cycle can be converted back to their corresponding amino acids through transamination reactions. Regulation of amino acid levels. Transamination reactions help regulate the levels of amino acids and keto acids within the cell, ensuring a balance between catabolism and anabolism. Intermediates for gluconeogenesis. During starvation or fasting, when glucose levels are low, some amino acids can be converted to intermediates of the TCA cycle, such as oxaloacetate and malate, which are then used for gluconeogenesis, the synthesis of glucose from non-carbohydrate sources. Specific examples of transamination reactions connected to the TCA cycle include alanine transaminase. This enzyme catalyzes the transfer of an amino group from alanine to alpha-ketoglutarate, forming pyruvate and glutamate. Pyruvate can then be converted to acetyl-CoA and enter the TCA cycle, while glutamate can be further metabolized. Aspartate transaminase, AST catalyzes the transfer of an amino group from aspartate to alpha-ketoglutarate, yielding oxaloacetate and glutamate. Oxaloacetate is a key intermediate in both the TCA cycle and gluconeogenesis. The amphibolic nature of the TCA cycle, its involvement in both catabolic and anabolic processes, is highlighted by its connection to transamination reactions. These reactions demonstrate the interconnectedness of metabolic pathways and how the TCA cycle plays a central role in coordinating energy production, biosynthesis, and the regulation of key cellular components. Cancer and TCA cycle. The relationship between cancer and the tricarboxylic acid, TCA, cycle, also known as the Krebs cycle or citric acid cycle, is complex and involves several key aspects. Metabolic shift in cancer cells. Cancer cells often exhibit a metabolic shift characterized by increased aerobic glycolysis and glutamine dependence, even in the presence of oxygen. This phenomenon, known as the Warburg effect, involves changes in TCA cycle activity and contributes to tumor growth. TCA cycle enzyme defects and cancer. Mutations in genes encoding TCA cycle enzymes are linked to various familial cancer syndromes. These mutations disrupt the normal functioning of the cycle, leading to metabolic imbalances that favor tumor development. Oncometabolites. Mutations in isocitrate dehydrogenase, IDH, and succinate dehydrogenase, SDH, result in the accumulation of 2-hydroxyglutarate, 2-HG, and succinate, respectively. These metabolites act as competitive inhibitors of various 2-oxyglutarate, alpha-ketoglutarate, and Fe2+ dependent dioxygenases, leading to alterations in cellular processes such as HIF-1 regulation and epigenetic modifications that promote tumorigenesis. Succinate's role in signaling and gene expression. Succinate acts as a signaling molecule by activating the SUCNR1 receptor, GPR91, a G-protein-coupled receptor involved in various physiological processes, including blood pressure regulation, immune responses, and angiogenesis. Additionally, succinyl-CoA, derived from the TCA cycle, participates in lysine succinylation (XUC) of histone proteins, an epigenetic modification that can influence gene expression. Nuclear TCA cycle enzymes and cancer. Several TCA cycle enzymes, including pyruvate dehydrogenase complex, PDHC, 2-oxyglutarate dehydrogenase complex, OGDC, and fumarate hydratase, FH, are also found in the nucleus. These enzymes play a role in nuclear metabolism and can be affected by mutations linked to cancer development. TCA cycle and inflammation. The TCA cycle is also involved in inflammatory responses, particularly in immune cells like macrophages. Itaconate, produced from the TCA cycle intermediate cisaconate, inhibits SDH and reduces ROS production, contributing to an anti-inflammatory effect. In summary, the TCA cycle is intricately linked to cancer development through various mechanisms, including metabolic reprogramming, oncometabolite accumulation, enzyme defects, altered signaling pathways, and epigenetic modifications. These connections highlight the importance of the TCA cycle as a potential target for cancer therapies. Location of the enzymes of TCA cycle. The enzymes of the TCA cycle are located in the mitochondrial matrix, with the exception of succinate dehydrogenase, which is embedded in the inner mitochondrial membrane. This localization allows for efficient coupling of the TCA cycle with the electron transport chain, which occurs in the inner mitochondrial membrane. Here is a breakdown of the locations of the TCA cycle enzymes. Citrate synthase, mitochondrial matrix. Aconitase, mitochondrial matrix. Isocitrate dehydrogenase, mitochondrial matrix. Alpha-ketoglutarate dehydrogenase complex, mitochondrial matrix. Succinyl-CoA synthetase, succinate thiokinase mitochondrial matrix. Succinate dehydrogenase, inner mitochondrial membrane. Fumarase, mitochondrial matrix. Malate dehydrogenase, mitochondrial matrix. The close proximity of these enzymes within the mitochondrial matrix facilitates the efficient transfer of intermediates between reactions, ensuring the smooth operation of the TCA cycle. Chemical inhibitors of TCA cycle. Chemical inhibitors of the TCA cycle. The TCA cycle, a vital metabolic pathway, can be disrupted by various chemical inhibitors. These inhibitors typically target specific enzymes within the cycle, leading to the accumulation of certain intermediates and disruption of energy production. Here is a breakdown of some key chemical inhibitors, their modes of action, and their sources. Fluoroacetate. Reaction inhibited. Conversion of citrate to isocitrate. Enzyme inhibited aconitase. Mechanism. Fluoroacetate is converted into fluoroacetate within the body. Fluoroacetate acts as a competitive inhibitor of aconitase, binding to the enzyme's active site and preventing the formation of isocitrate. Sources. Fluoroacetate is a naturally occurring toxin found in some plants. It is also used as a rodenticide. Arsenite. Reaction inhibited. Conversion of alpha-ketoglutarate to succinyl-CoA. Enzyme inhibited. Alpha-ketoglutarate dehydrogenase complex. Mechanism. Arsenite binds to the dihydrolipoamide cofactor of the alpha-ketoglutarate dehydrogenase complex, inhibiting its activity and leading to the accumulation of alpha-ketoglutarate. Sources. Arsenite is a toxic compound found in some insecticides and herbicides. It can also be a byproduct of industrial processes. Malonate. Reaction inhibited. Conversion of succinate to fumarate. Enzyme inhibited. Succinate dehydrogenase. Mechanism. Malonate is a structural analog of succinate and acts as a competitive inhibitor of succinate dehydrogenase. It binds to the enzyme's active site, preventing succinate from binding and being oxidized. Sources. Malonate is a dicarboxylic acid found in some plants. It is also used in laboratory settings to study the TCA cycle. In addition to these three main inhibitors, other chemicals can disrupt the TCA cycle. For instance, rotenone and amobarbital inhibit NADH dehydrogenase, complex I of the electron transport chain, indirectly affecting the TCA cycle by disrupting the reoxidation of NADH. Similarly, antimycin A inhibits cytochrome B in complex 3 of the electron transport chain.